Okay, so how do we tell if a SNP is being caused by natural selection or drift? So when I say being caused by, I mean, why is it increasing in proportion in the population is it due to natural selection or due to drift? And so one of the things that we could look at is the proportion of the population that carries this SNP. So for example, I just made up a position in a genome here, and in this part of the genome, originally in the ancestral state, there was a G, and so maybe at time 0, most of the population had a G in this position 1640, and so we'll say it's like 80, 90% up here, and then very few individuals had an A at this position. That was a mutation, but we know that some individuals do have that. But then perhaps we see over time that the proportion of individuals with that A is increasing, while the proportion of the individuals with the G or the ancestral state is decreasing. And then maybe we even see that the number of individuals with A goes to fixation, and the number of individuals with the G goes to 0. So fixation is something that we inferred or kind of didn't quite talk about, but we saw yesterday. Fixation is when there's no diversity at a certain site in the genome. So fixation is equal to no diversity. In other words, every individual is the same. And so we talked about how that can be disadvantageous for a population, because then there's no variety for if a new environmental pressure comes up, then there's no variety or diversity to select on. But we see this sometimes because sometimes there's just such an advantage to having a certain SNP or other type of mutation that the whole population is then going to end up having that. So in this example, we used SNP, but we might have easily done this for the horizontal gene transfer that I spoke about in another video. And in that case, we definitely do expect that to go to fixation. We expect all bacterial individuals in a population to transfer or to receive that transferred set of genes that gives them resistance to antibiotics, because that can happen fairly quickly and is extremely advantageous. So we expect to see it go to fixation. So this is a piece of data that might suggest that natural selection is working here, and that this is not just due to drift. However, we know that this can happen due to drift if there's small population sizes, if there are things like migration occurring either into or out of the population, bottlenecks, etc. So something else we can do to look at or to determine whether this is caused by natural selection or drift, that is the increase of a certain mutation in the population being caused by natural selection or drift, is to look at the phenotype, right? So we could try and correlate, we could pretty easily do that with the example of antibiotic resistance. We see the phenotype of being resistant to antibiotics. Bacteria will have that. That's obviously, you can do an experiment in a lab showing that that phenotype is advantageous. But maybe we're looking at an organism, a multicellular organism that is much more complex. And maybe it's not so easy to tell whether or not this SNP makes a difference in a phenotype, or what phenotype it makes a difference in, or under what environmental conditions. Or maybe we're looking at an organism that's extinct, so we don't have that much phenotypic data. And so in that case, we can look at some other things. So let's stop this share, and I'm going to share my screen. Yes. And in that case, we want to look at some other stuff. So again, we're looking, in this case, we're looking at SNPs in particular, not large mutations like the gene transfer. And we want to look at synonymous versus non-synonymous SNPs or mutations. So again, that's synonymous versus non-synonymous. We'll get to that in a minute. And here I always advise people to look at the root of the word, the Latin root. And in this case, it does help. So if we look at synonymous, what word does that seem similar to? It's similar to the word synonym. What is a synonym? A synonym is a word that is analogous or equal to another word. You could change them out. They mean the same thing, right? And so a synonymous mutation is one that is equal. I like to think of it that way. That is equal to another gene sequence. So I'm going to write that right here. Equal sequences. Now, I don't mean equal in the number of base pairs or something. I mean, I do, but non-synonymous would also be equal in the number of base pairs. I mean equal in their meaning. So just like synonyms, words that are equal in their meaning, we're referring here to sequences that are equal in their meaning. So if we are looking at these two codons, they are different by one base pair. So this could be the cause of, or the result of a SNP, right? Some mutation. So in the original, which I'll call A here on the left, we had an ACU, and that codes for the amino acid threonine during translation. In the SNP, we had some mutation that caused this codon to change the last base pair here to ACA. This also codes for threonine, the amino acid, during translation. So these two sequences are quote unquote equal in their meaning. They code for the same amino acid, and they are synonymous. So this is a synonymous mutation. It does not change the amino acid. I guess you could kind of guess at this point what a non-synonymous mutation is. This means that the mutation is not equal. It codes for a different amino acid. And so here we see the first sequence, A, is GGA. This codes for a glycine amino acid during translation. And then in B, we've had some type of mutation, which causes a SNP. The changed sequence is AGA, and this no longer codes for glycine. This codes for arginine during translation. So these are obviously not equal. They're not the same amino acid. Take a step back for a minute and consider which amino acids we changed in these examples. And this was done for a reason. For the synonymous mutation, we changed the third we changed the third base pair. In the non-synonymous mutation, we changed the first base pair. Why is that? Think for a moment, and you might recall that there's something called the wobble base. The wobble base is the third nucleotide in a codon. And for many amino acids, it doesn't matter what the third base pair is. So for example, you might have ACA, ACG, ACC, etc. and it would code for the same amino acid. That third nucleotide does not matter. Again, I'm not sure if it's exactly, if it's specifically threonine has that characteristic. But then when we look over here for the non-synonymous mutation, we change the first base pair. That's not the wobble base. Obviously, it's not the third amino or the third nucleotide in the codon, sorry. It is the first codon or the first nucleotide in the codon, and therefore it changes the amino acid. Now the wobble base sometimes does matter, but the third base pair is less likely to matter because of the redundancy of the genetic code, or in other words, because of the wobble base. Now, one thing that I also wanted to mention here, which is not in your textbook, so pay attention here, all hands on deck. These two things that I've written up here. So DS and DN. DS is for synonymous, DN is for non-synonymous. And this is how you would actually use the data that you got. You can use this equation. You would look at the change in non-synonymous mutations versus the change in synonymous mutations. And you could do this if you have perhaps 100 individuals that you sampled from, and you compare their gene sequences for a particular gene, and you look at, maybe you look at all of the SNPs, or maybe you look at the SNPs at a certain position in the gene. And if you're seeing significantly more non-synonymous mutations than synonymous mutations, then you're more likely, or this is more likely, you're more likely to conclude that this is the effect of natural selection. Because what essentially you're comparing here are the number of mutations, or SNPs in particular, that occur accidentally, which would be the synonymous mutations. You could think of it that way. These occur accidentally, but they don't matter, so they're not fixed, versus the non-synonymous mutations, which, yeah, these can originate accidentally, but they do matter. They are more likely to affect the protein because they're affecting the amino acid sequence. So once you change an amino acid in a protein, you're more likely to change its shape, which means you're more likely to change its function. So again, you can't just look at a single DNA sequence from a single organism and say, oh, yes, this is a synonymous SNP. No, you need to compare at least 2 sequences, usually as many as possible, to see if these are synonymous versus non-synonymous mutations. And you could, again, start to get a feel for whether or not this is natural selection, if there are more non-synonymous mutations, or if this is drift, and that would be more synonymous mutations.